Okay. Um, in this chapter, we're going to touch briefly on genomics and proteomics, okay, which are methods utilized today to both manipulate and analyze entire genomes and the expression of entire genomes. And we start out with a picture of yeast, okay? In the spring of 2014, a group of scientists reported the first synthesis of a completely artificial eukaryotic chromosome. When substituted for chromosome 3 in Saccharomyces cerevisiae, shown here, the synthetic chromosome functioned well. Remarkably, much of the work in constructing the artificial chromosome was carried out by undergraduate students at Johns Hopkins who completed the task as part of their Build a Genome class. Saccharomyces, commonly known as baker's yeast, has been used for thousands of years for making bread and beer, and today it's important in the production of food and chemicals. It's also an important model genetic organism. Baker's yeast is a single-celled eukaryote. It's easy to cross and grow in the lab, and it was the first eukaryote to have its genome completely sequenced, and a number of molecular techniques have been developed to manipulate its DNA. Scientists are now working to create a completely synthetic genome for Saccharomyces, which is a flexible genome that can be altered at will to produce yeast with selected genes and special properties. The first step in creating a synthetic genome for yeast was the synthesis of an artificial chromosome known as artificial chromosome 3. Scientists began by designing the new chromosome on the computer using the basic sequence of a natural chromosome 3 but deleting non-essential DNA, including introns, repeated sequences, near telomeres, transposable elements, and transfer RNA genes. They designed the artificial chromosome with special sequences that allow easy deletion of genes and rearrangement of the chromosome, of the genome rather. They also changed all the TAG stop codons to TAA, freeing up the TAG codon for future use in encoding an additional artificial amino acid. Once the sequence of the chromosome was designed, the actual construction began. As a first step, the students in this class stitched together small pieces of DNA in 750 base pair building blocks. Each student started with a series of overlapping 80 base pair DNA fragments called oligos, which were synthesized on a machine and provided the class by a commercial firm. The students assembled these fragments into building blocks using methods based on PCR. After verifying that the correct sequences were joined together, the students cloned the building blocks into bacteria to make many copies. Scientists then assembled the building blocks into 127 larger overlapping fragments of about 7,000 base pairs, or 7 kilobases. These were called mini chunks. And finally, they systematically replaced the native sequence of the yeast chromosome 3 with the mini chunks of artificial DNA until a completely synthetic chromosome was produced. The resulting artificial chromosome, which is dubbed SYN3, consists of about 300,000 base pairs of DNA, replacing the original 316,000 base pairs of the normal chromosome. When inserted into yeast cells in place of the normal 3, it functioned normally, replicating and expressing its genetic instructions. Cells with SYN3 had the same morphology as a natural yeast cell and grew well. They appeared to have no fitness differences between SYN3 cells and natural yeast, and the genomes of the SYN3s were stable for over 125 mitotic divisions. In the design of SYN3, geneticists inserted special sequences called LOXP-SIM sites, which are on either side of many of the genes, providing an easy way to create variations of the chromosome by deleting and rearranging genes. The LOXP-SIM sites are recognized by an enzyme called Cre, which when activated by a chemical added by the researcher, brings about recombination between the LOXP-SIM sites, resulting in deletions and rearrangements of the genes. This method of changing the genome is called synthetic chromosome rearrangement and modification by LOXP-mediated evolution, or SCRAMBLE for short. One reason scientists wanted to create an artificial yeast genome is so they could systematically take out groups of genes to determine the minimal genome necessary for a cell to function. The SCRAMBLE system greatly facilitated this effort. Perhaps the greatest impact of the project was on the students who helped to advance the field of genetics. while taking an undergraduate course. In a now expanded effort called Synthetic Yeast Genome Project, additional students and scientists from schools around the world are synthesizing the other 15 chromosomes in yeast with the goal of creating a synthetic genome and thus a synthetic organism. Okay, so this is something that wouldn't have been possible as recently as a decade ago. Okay, um, the way that this was brought about was through advances in the production of polynucleotides on a solid state surface, okay, and through advances in DNA sequencing, okay. Structural genomics is the study of the organization and sequence of genetic information contained within a genome, providing the basic DNA sequence that's used in functional and evolutionary studies. An early step in characterizing a genome may be to prepare genetic and physical maps of the chromosomes, which provide information about the relative location of genes, molecular markers, chromosome segments, often essential for positioning chromosome segments and aligning stretches of sequenced DNA into a whole genome sequence. These maps are also the foundation for positional cloning, in which genes that influence specific traits are located and sequenced. Genetic maps, sometimes called linkage maps, provide a rough approximation of the location of genes relative to other known genes. These maps are based on genetic process of recombination. The basic principles of constructing genetic maps are going to be discussed, were already discussed in a previous chapter. In short, individual organisms of known genotype are crossed, and the frequency of recombination between loci is determined by examining progeny. If the recombination frequency between two loci is 50%, and the loci are either on different chromosomes or far apart on the same chromosome. If the recombination frequency is less than 50%, the loci are close together on the same chromosome or may in fact be the same gene. For linked genes, the rate of recombination is proportional to physical distance between the loci. Distances on genetic maps are measured in recombination frequencies known as centimorgans or map units. Data from multiple 2-point or 3-point crosses can be integrated into linkage maps for whole chromosomes. For many years, genes could be detected only by observing their influence on a trait, that being the phenotype, and the construction of genetic maps was limited by the availability of single locus traits that could be examined for evidence of recombination. Eventually, this limitation was overcome by the development of molecular techniques, such as the analysis of restriction sites and the development of PCR and DNA sequencing, which are able to provide molecular markers that can be used to construct and refine genetic maps. There are limitations to genetic maps, however, the first of which is the resolution or the detail. The human genome includes 3.2 billion base pairs of DNA and has a total genetic distance of 4,000 centimorgans, an average of 800,000 base pairs per centimorgan. Even if a marker were present in every centimorgan, their resolution in regard to their physical structure would still be low. In other words, the detail of a genetic map is limited. A second problem with these maps is that they don't always accurately correspond to physical distance between genes. Genetic maps are based on rates of crossing over, which vary from one part of the chromosome to another, so the distance in a genetic map is an approximation. Figure 20.2, which comes next, compares the genetic map of chromosome 3 in yeast with the physical map determined by DNA sequencing. There are some discrepancies in the distances between genes and even in the position of some genes. In spite of these limitations, genetic and physical maps and the sequencing of whole genomes provide valuable information. You can see it here. Okay? So notice the difference. Notice the difference, right? Genetic map, and then we have the bands visible on the metaphase chromosome, right? And then if we compare that to what we call the physical map, we get an even more accurate depiction of the relative location of these genes. Genetic maps have limitation in terms of detail. The human genome has 3.2 billion base pairs of DNA and a total genetic distance of 4,000 centimorgans, okay? And as we talked about, the rate of crossing over varies between different regions of the chromosome, right? So what you're looking at here is chromosome 3, and you can see the difference between the distances on the physical and the genetic map, Okay. Physical maps are based on direct analysis of DNA, and they put genes in relation to distances measured in a number of base pairs, kilobases, or megabases. A common type of physical map is one that connects isolated pieces of genomic DNA that have been cloned in bacteria or yeast. Physical maps generally have higher resolution and are more accurate than genetic maps. A physical map is analogous to a neighborhood map that shows the location of every house along a street, while a genetic map is like a highway map that shows general locations of major towns and cities. One of the techniques that's been used for creating a physical map is restriction mapping, which determines positions of restriction sites in DNA. When a piece of DNA is cut with a restriction enzyme and the fragments are separated on a gel electrophoresis apparatus, the number of sites in the DNA and the distance between them can be determined by the number and the positions and bands on the gel. However, this information doesn't tell us the order or the precise location of the restriction site. To map the site, a sample of DNA is cut with one restriction enzyme, and another sample of same DNA is cut with a different one. A third sample is cut with both. The DNA fragments produced by these digests are then separated on the gel, and their sizes are compared. Overlap in the sizes of the fragments produced by the digest can be used to position the restriction sites. Okay? Most restriction mapping is done with several restriction enzymes used alone and in various combinations, and produces restriction fragments that can then be pieced back together. With long pieces of DNA, computer programs are used to determine the restriction map, and restriction mapping may be facilitated by labeling one end of a large DNA fragment with a radioactive tag and identifying the end using something called a probe, which is an oligonucleotide that has either a radioactive isotope in it or a dye that can fluoresce when hit with a laser of a particular wavelength. By 80, methods for mapping and sequencing DNA were sufficiently developed that the geneticists began to propose that the entire human genome be sequenced. So this was the Human Genome Project. Estimates suggested that 15 years and 3 billion would be required to do this. The Human Genome Project began in October of 90. Initial efforts were on developing new automated methods for cloning and sequencing DNA and generating detailed physical and genetic maps of human DNA. The effort was a public project consisting of international collaboration among 20 research groups and hundreds of individual researchers who formed the International Human Genome Sequencing Consortium. The group used map-based strategies for sequencing the human genome. So what are these map-based strategies? Short sequence fragments are assembled into a whole genome sequence by first creating a detailed genetic and physical map of the genome, which provides known locations of genetic markers, for instance, restriction sites or known DNA sequences, at regularly spaced intervals along each chromosome. These markers are later used to help align the shortened sequence fragments in the correct order. Okay, so you get landmarks, you overlap the landmarks, and you make what we call the contig. Okay, which is the assembled fragment. Okay. The first genomes to be sequenced were small virus genomes. The genome of bacteriophage gamma, consisting of 49,000 base pairs, was completed in 82. In 95, the first genome of a free-living organism, Haemophilus influenza, a bacteria, was sequenced by Venter and Fraser of the Institute for Genomic Research, known as TIGER, and Hamilton and Smith of Johns Hopkins. This bacteria has a small genome of 1.8 million base pairs. By 96, the genome of the first eukaryotic organism, yeast, had been determined, followed by the genomes of E. coli in 97, C. elegans in 98, and Drosophila in 2000. Arabidopsis followed in 2000. Sequencing of the human genome was completed in 03. Once the genetic and physical maps are available, chromosomes or large pieces of chromosomes are separated by pulse field gel electrophoresis or flow cytometry. Standard gel electrophoresis can't separate the large pieces of DNA, such as whole chromosomes, but PFGE can separate large molecules of DNA or whole chromosomes in a gel by changing the orientation of the electrical current. So basically what happens is that you generate a current perpendicular, then parallel, then at an angle to the chromosome fragments, so that you pull and you snap them in different directions and you affect their separation. Okay. In flow cytometry, chromosomes are sorted optically by size. Each chromosome is then cut up by partial digestion with a restriction enzyme, which means that the enzymes are allowed to act for a limited time so that not all the sites are cut. The partial digestion produces large sets of overlapping DNA fragments, which are cloned with the use of plasmids, or yeast artificial chromosomes, or bacterial artificial chromosomes. Next, these large insert clones are put together in their correct order on the chromosome. This assembly can be done in several ways. One method relies on the availability of high-density map of genetic markers. A complementary DNA probe is made for each genetic marker, and a library of the large insert clone is screened with the probe, which hybridizes to any colony that has a clone with a marker. The library is then screened for neighboring markers. Because the clones are much larger than the markers used as probes, some clones will have more than one marker. As an example, clone A might have markers M1 and 2, clone B might have markers M2, 3, and 4, and C might have markers 4 and 5. Such a result would indicate that these clones contain overlap, and it will help you generate the contig rather. Okay, so that's shown here, right? In '98, Ventnor announced that he would lead a company called Celera Genomics in a private effort to sequence the human genome. He proposed using a shotgun sequencing approach in which we essentially shatter the genome and utilize oligos in order to generate sequence information. Okay. In whole genome shotgun sequencing, small insert clones are prepped directly from human DNA and are sequenced. Computer programs then assemble the entire genome by examining the overlap of the small insert clones. One advantage of the shotgun is that the small insert clones can be placed in plasmids, which are simple and easy to manipulate. The requirement for overlap means that most of the genome must be sequenced many times. The average number of times a nucleotide in the genome is sequenced is called the sequencing coverage. As an example, 10 times coverage means that an average nucleotide in the genome has been sequenced 10 times. In the summer of 2000, both public and private sequencing projects announced the completion of a rough draft that included most of the sequence of the human genome, 5 years ahead of schedule. The human genome sequence was declared completed in the spring of '03, with some gaps still remaining. For most chromosomes, the finished sequence is 99.99% accurate with less than one base pair error per 100,000, an accuracy rate 10 times that of the initial goal. With the first human genome determined, sequencing additional human genomes is a lot easier. It's now possible to sequence an entire human genome in a few hours, and genomes from thousands of different people have been sequenced. The cost of sequencing a complete human genome has also dropped dramatically and will continue to fall as sequencing technology improves continuously. Genome sequencing has identified a number of sites where humans differ in their genome sequences. Imagine that you're riding in an elevator with a random stranger. How much of your genome do you have in common with that person? Studies of variation in human genomes indicate that you and that stranger will be identical 99.9% of your DNA. Okay? So that's called your homology. The difference between you and the stranger is very small in relative terms, but because the human genome is so large, you and the stranger will be different at about 3 million base pairs of your genomic DNA. These differences are what make each of us unique, and they greatly affect our physical features, our health, our intelligence, and our personality. A site in the genome where individual members of a species differ in a single base is called a single nucleotide polymorphism, or SNP. Arising through mutation, SNPs are inherited as allelic variants in the same way as alleles are produced in phenotypic differences. Although SNPs don't usually produce a phenotypic difference, okay? They're usually changes in regions of the DNA that may be highly repeated, may not, but aren't often coding, okay? In a comparison of the same chromosomes from two different people, a SNP can be found in approximately every 1,000 base pairs, or about every kilobase. SNPs within a haplotype are physically linked and therefore tend to be inherited together. New haplotypes can arise through mutation or crossing over, which breaks up the particular SNPs in a haplotype. because the rate of crossing over is proportional to the physical distance between the genes. The SNPs and other genetic variants that are located close together on a chromosome will be strongly associated as haplotypes. The non-random association between genetic variants within a haplotype is called a linkage disequilibrium. Because SNPs and a haplotype are inherited together, a haplotype consisting of thousands of SNPs can be identified through the use of only a few. The few SNPs used to identify a haplotype are called tag SNPs. There are about 10 million SNPs in the human population, but because of linkage disequilibrium, the SNPs can be grouped into much smaller numbers of haplotypes. Therefore, a relatively small number of SNPs, perhaps only 100,000, can be used to identify most of the haplotypes of humans. So what are these? These are just areas, common areas in DNA shared between individuals of a species that will have a base or two change. Okay? And we can track their heritability the same way that we could track a trait, right? A physical trait back in Mendel's days. We now have the ability to see this trait through the use of DNA sequencing. Okay? So essentially, because of the change in instrumentation, what we've been able to do here is now create a group of traits that before that instrumentation was produced were not visible, were not characterizable, and were not able to be assigned by inheritance. Okay? So this gives us another tool in order to organize genes and understand the arrangement of genes on a chromosome and within the genome. Okay? Most SNPs present within a population from a single mutation that occurred in a particular chromosome in a particular individual and then spread throughout the population. Each SNP is initially associated with other SNPs that were present on the particular chromosome on which the mutation came. The specific set of SNPs and other genetic variants observed on a single chromosome or part of a chromosome is what we call a haplotype. Okay? So there is an example of a haplotype, right? You got a chromosome, okay? Same chromosome, chromosome number 1, right? Different individuals, A, B, C, and D, all right? Basically the same information, but tiny changes, okay? And do these changes lead to a change in phenotype? They may not, but what we use them for is to determine the locations of genes that do produce changes in phenotype. So we call them markers, okay? A diploid person normally possesses 2 copies of every gene, one inherited from the mother and one from the father. Nevertheless, studies of the human genome have revealed differences among people in the number of copies of large DNA sequences, and these variants are called copy number variations. Complete genome sequences have now seen or determined numerous organisms and many additional projects underway to determine the location of these differences in DNA sequences. These studies are producing a lot of sequence data. Cataloging, storing, and retrieving and analyzing this data is a big challenge in modern genetics. Bioinformatics is an interdisciplinary field that combines molecular biology and computer science, and it centers on the development of databases, computer search algorithms, gene prediction software, and other analytical tools that are used to make sense of DNA, RNA, and protein sequence data. Bioinformatics develops and applies these tools to mine the data, that is, to extract the useful information from the sequencing projects. The development and the use of algorithms and computer software for analyzing sequence data have helped to make molecular biology a quantitative field. The storage of sequence data in publicly available online databases allows scientists and students throughout the world to access this when they do their work. Okay? So what's happening here, okay, by examining in great detail DNA sequence, is that we're discovering landmarks and characteristics that we can watch in terms of heritability and that we can ask questions about in terms of, are they near an expressed region of the DNA, an EST, right? A marker associated with DNA sequences that are expressed as RNA, okay? Will that RNA encode a protein? It may not, okay? But the bottom line is that this is beginning to give us an outline or a shape of what the genome looks like. Advances in sequencing technology, which have made sequencing faster and less expensive, provide the possibility of sequencing not just the genomes of individual species, but the genomes of entire communities of organisms. Metagenomics is the emerging field in which the genome sequences of an entire group of organisms that inhabit a common environment can be sampled and determined. Our ability to sequence and study whole genomes, coupled with our increased understanding of the genetic information required for biological processes, provides the possibility of creating entirely from scratch novel organisms that have never existed. Synthetic biology is the field that seeks to design organisms that provide useful function, such as microbes that clean energy or break down toxic waste, or produce byproducts that have an important market, such as a particular drug, or may carry out a particular function in an industrial application, okay, such as yeast designed to produce things like beer or bread more effectively, okay? So we're learning from really getting down to the nitty gritty here, what the base sequence is, um, what the arrangement of information is in the organism, and we can use that to compare that information to other organisms and determine things like evolutionary trees, okay, and to impute potential functions or to design new functions for existing genes, okay? So this is a fast evolving area of biological science that requires training in the use of a wide variety of computer programs and the ability to understand and interpret genetic data. Okay. All right. I will join everybody in the next podcast. I thank you for listening.